Okay, thank you. Hi, my name is Erika. I'm a bioinformatician at Mount Sinai Hospital in Toronto, and today I'll be talking to you briefly about the Canadian Genomic Data Commons, or CGDC project. Oops, let me just zoom in. How do I? Sorry, how do I switch the? It's a little confusing here.
Just here in this screen is two screens.
Oh, okay, perfect. Thank you. Okay, so today we'll just be going through some of the objectives and goals of the Canadian Genomic Data Commons project, which is a 5-year Canadian Foundation for Innovation funded project. I'll be discussing some of the plans for the sharing of genomic data through the Canadian Open Genetics Repository, or COGER activity, generating allele frequency data for Nomad Canada, talk about the launch of a Canadian instance of Seeker at Sick Kids Hospital, and also just really briefly touch on the CGDC's overlap with some of the planned activities for the PCGL and All for One projects. Oops, skipped ahead. So this is our leadership team. So it's led by Dr. Jordan Lerner-Ellis at Mount Sinai Hospital, as well as Dr. Kim Boycott at the University of Ottawa. But we have representation from across the country and from many of our other team members, as well as some international representation as well from the Broad Institute. And so start off, basically in the next few years, there's going to be tens of millions of genomes sequenced in healthcare. And this is going to be an extremely valuable resource for us. But yet a lot of this data in healthcare has still been largely inaccessible for researchers. And so the basis for the CGDC is that we really want to create a new digital infrastructure in order to transfer genomic data from healthcare into a new national data sharing research ecosystem for Canada, which will be especially important in the cases of rare disease, which inflict some 3 million Canadians today. And so this activity is broken down into 3 major branches or activities. The first of which is COGER. So the Canadian Open Genetic Repository was originally established in 2012, and it was a unified open access or centralized genomic database for standardizing variants and sharing these variant interpretations across Canadian diagnostic laboratories. It had 22 labs participating with some 50,000 variants in the database. And so with the CGDC project, we now want to expand on the functionality of the original COGER database by deploying new lab-specific pipelines to collect data from of these other diagnostic labs across the country, export these interpretations, develop new options for data sharing, as well as integrate discrepancy reporting in order to alert labs, likely by email notification, if there are discordancies in terms of their variant interpretations and resolve those. And also expand this functionality out for somatic and CNV data as well. For the second activity, Nomad Canada. So Nomad, as you know, it's an international coalition to aggregate and harmonize whole genome sequencing data and summarize allele frequencies. But as we know, it's been predominantly European up until recently. And so with Nomad Canada, we want to build out a Canadian version of the Nomad database with genomes from 50,000 genomes from other large-scale and clinical sites across Canada, which will be especially important for underrepresented populations, as we heard of this morning, such as Indigenous Canadians. And also develop new strategies to harmonize and display those results in a Canadian-hosted Nomad browser. And as part of Activity 3, Tools for Rare Disease, we want to deploy the Seeker software on HPC for health infrastructure starting in Sick Kids Toronto. So really briefly, Seeker is a user-friendly software that can help to facilitate the identification of disease-causing variants. And deploying this at Sick Kids would give a way for researchers across Canada to facilitate new gene disease discovery for research. And then secondly, we also want to assist in the building out of an e-consenting portal for use with different demonstration projects happening today, like Care for Rare. And this would essentially be a participant portal that they could go into to adjust their consents to some of these various projects, see all the studies that use their data or studies that they might be eligible for enrolling in, and potentially update some very basic health info. So jumping into the first activity, COGER. Again, the goal here is that we want to create a unified, open access, clinical-grade genetic database for Canadian diagnostics labs to use and to put all of their data on a commonly shared platform. So to date, we already have 30 different Coder collaborators spread across the country. I know it's hard to see all of them. There's quite a few, 8 of which are from the All for One sites. All for One, again, is a project. It's 6 different clinical implementation projects also happening in Canada. And so essentially, we want to, this is sort of how the workflow for Coder might look. So a lot of these labs currently have data in either a laboratory information system or a commercial software. And we at Sign, as the core facility, would be working with these labs to help them to export their raw data and run custom pre-processing scripts to clean that data, to filter it, to gather just the information we want to put into the database, and then transfer that data into the major unified Coder database alongside the data from other various sites as well. And then we would like to build out discrepancy or consensus building tools in order to have variant discrepancy assessments. So again, these labs will be alerted if there is a discrepancy reported in their interpretations. They can then work on resolving that issue so that we end up with a curated consensus database for Coder. And this would happen periodically. And then that database, again, periodically would be pushed to other public access databases like ClinVar. So again, just to show you what this would look like, again, these labs across Canada would be pushing their clinically validated, de-identified data into this main repository. They would have read-only access to view and validate their data with other labs for discrepancy reporting. And then periodically, once curated, that data would be pushed to other available databases like ClinVar Online. Jumping into our activity 2, Nomad Canada. Nomad Canada aims to be a publicly available variant database for genome sequencing with summary data on allele frequencies as well as phenotypic info. But Nomad already exists, so why do we need a Canadian database? And the reason for that is that there's a number of large-scale genomic projects already happening in Canada with a lot of global impact. So for example, HOSEEK, this was a project with 10,000 genomes sequenced that was focused on the genomic architecture to host response and disease for COVID-19 affected individuals. And this is actually going to be Nomad Canada's pilot data set. We're currently working on this. All for One, as I just mentioned, Coder, Care for Rare, which focuses on rare disease, as well as ICGC Argo, the Missing Whole Genome Sequencing Project, and MOHCCN. So lots happening. And so essentially here we want to build on the model of the Broad's version 3 and 4 releases of Nomad, with the hopes that this will increase the ancestral diversity of Nomad through the inclusion of these underrepresented populations. We also want Nomad Canada specific features, so the integration of phenotypic info, and eventually create a user interface that will be shared publicly on both a Canadian hosted web browser and the Broad's Nomad browser as well. But of course, there's a few bioinformatics and technical challenges associated with this. One of the big problems is that not all this data can be shared or combined in a single location. So a lot of the project consent for these don't allow for physically moving the data. Much of this data is hosted across multiple different cloud computing resources, as well as HPCs, so all over the place. Some of the data may need to be reprocessed for in-pipeline harmonization. And the All for One project specifically, they actually only allow for the sharing of their aggregate data. So we wouldn't necessarily have access to their raw data in order to run the Nomad code. They would only be able to share like their summary statistics at the end. So some challenges. But to start off, we would essentially start by processing whole genome sequencing data from the 50,000 individuals from these participating sites where possible. So again, for large scale projects where we can, we would love to be able to joint call that data and then run the Nomad code to generate these Hale matrix tables, the summary statistics that include the allele frequencies, feed that into a variant database that can then be uploaded to an eventual Nomad Canada browser. For the All for One sites, which again, they have more heavy data sharing restrictions, we would work with their sites to help facilitate the running of their own Nomad code, wherever it's being stored on their end. And then they can just share a subset of their summary statistics at the very end, which can be hopefully combined with the existing variant database and then uploaded to the browser as well. And as we heard this morning, we would also love to create linkages to the Silent Genomes project as well, where possible. So obviously, if we can't combine all the data, that gives issues to the issue of estimating genetic ancestry and relatedness across these samples coming from different sites. But there are ways to circumvent these data sharing restrictions. So one method that we've been thinking about is using PCA. So we would ask each participating site to perform a PCA analysis of their individuals, and then project those PCs against the Nomad3 reference panel. And then those projected PCs can be shared. And theoretically, if the same projected PC is identical in, say, 2 separate studies, it might be assumed that that individual is maybe identical or related in some manner, which would assist us in our quality metrics and ensuring that that's not an issue for downstream. So even though it is a challenge, we can circumvent these data sharing restrictions, and we'll be working on other methods to do this as well. And then as a new feature for Nomad Canada, which doesn't exist in the current browser, is that we would love to be able to integrate phenotypic information through the use of ICD or HPO codes. And the reason is that some population databases like Nomad, they're often used as controls for interpreting variants, but these databases can not only be assumed to include healthy individuals. And so with this, researchers could essentially define their own controls by displaying the proportion of individuals for each ICD or HPO term per variant. Jumping now into our activity 3. So again, Seeker, essentially, as I mentioned before, we'd love to be able to deploy Seeker on HPC for Health infrastructure in sick kids and make that available for researchers. So Seeker is an open source web-based tool developed by the Broad Institute for Rare Disease Genomics, and it allows researchers to be able to search and annotate their genomic data sets. There's already some 2,000 people from 100 plus countries that have family-based analysis in Seeker. It's been extremely helpful for different gene disease discoveries within the platform. And there's many new features planned for 2024 and beyond, like RNA-seq analysis. And so our steps for activity 3 are: 1, we want to deploy, again, this software on HPC for Health infrastructure in sick kids, and then use a demonstration for Seeker using data from the Care for Rare and Genomics for RD projects. So really briefly, Care for Rare is going to be a 10-plus year disease research program from 20-plus clinical genetic sites in the country. And Genomics for RD is the infrastructure being used to support the Care for Rare projects. And these 2 steps, I'm happy to say, are already completed, actually. After the initial deployment, we want to load data for 3,500 exomes from Care for Rare into Seeker, and then expand that functionality out for Seeker to include another 10,000 exomes from other rare disease cohorts as well. And then step 4, facilitate data sharing by enabling other researchers in clinical labs to participate within Seeker in the Matchmaker Exchange, which is a federated network of global databases that enable rare disease and gene discovery. So again, as I mentioned, this has already been completed. The Center for Computational Medicine already has a local instance of Seeker installed that's publicly available for any researchers to check out. And it's already been utilized by groups at Sick Kids and Care for Rare, as I mentioned. And then I also just want to touch briefly about how the CGDC project will overlap with some of the other projects happening. So again, All for One, it's made up of 6 different genome implementation projects across Canada with 10,000 genomes planned for sequencing. And it has a clinical arm, which is a national network of diagnostic labs, and a research arm, which is the most important for us, which is a national registry for participants that want to be contacted about their research. And so the CGDC will be assisting and working with the All for One team to help develop out their e-consenting platform. As I mentioned, this would be a dynamic consenting platform for participants, again, so that they can go in, adjust their consent, see all the studies that they're part of and eligible for, maybe update some basic health info, things like that. And also the data collected as part of the research arm will be used to build it as demonstration for the Nomad Canada and Seeker projects as well. And also just like to highlight how a lot of this also overlaps with the PCGL projects as well. So again, they're also working on similar things in terms of building out a participant portal for this dynamic consent, as well as hopefully integrating some of the data collected from the research arm of All for One into the eventual PCGL database. And then just finally, I'll just touch on how the CGDC overlaps with the PCGL as well. So again, we'll be working really closely with the team as some of our goals align with in terms of data processing, for example, joint calling and harmonizing some of this data potentially for CGDC, or at least making sure it's all harmonized and has sequence on the right dragon and other parameters and QC. We'll be using some of their clinical data, of course, for maybe the integration of their phenotypes into Nomad Canada. building out the participant portal, as I just mentioned. We'll be working really heavily with their team to work on that, as well as eventual creating integrations and linkages from Nomad Canada into the Variant Store browser. And with that, I just want to say thanks. This is our team at Mount Sinai. We're all contributing heavily to this project. And that's everything from me.
Thank you, Erika. We have time for a few questions.
Hello, thank you so much for the talk. It's a great news that Canada is moving forward with Nomad Canada. As you mentioned, to obtain a high-quality data set, we'll have to recall variants, right? We'll have to get the samples together and recall variants, at least for these 50,000 whole genome samples. 10 years ago in Nomad, they've done it with JTK because the Dragon wasn't available then, but now it's a completely different landscape mathematically. So are you planning to use Dragon for this project, or will it be JTK-based calling?
That's a great question. So I will say we're trying our best to make sure that we're aligned with what the Broad is doing for all these different versions of Nomad. I think they're almost ready to push their version 5 release out, which is using Dragon. So however closely that we can align our Nomad Canada with what the Broad's plans are, because we would like to also host that data within the Broad's instance of Nomad as well. We'll try to align those goals very similarly. So I would say we would try to move forward with Dragon as much as possible. But of course, that can pose some issues if we have to reprocess data. But where possible, we would like to use the most up-to-date pipelines for the Broad and beyond.
Hi. I was wondering, the discrepancy tools that you mentioned, would it just be an alerting mechanism, or would there be an ability for labs to go in and kind of check how the variant calling happened? I imagine different labs are going to be calling variants in different ways. And I was wondering how that would be tackled.
That's a great question. So I think that's still in discussion, and a lot of these tools are still being in the process of built out. So it's definitely a great question. I don't think it's determined yet what sort of data will be in the to the extent of data about the variant interpretations in the database, but hopefully we'll be able to have some sort of indication of what ACMG criteria was used, what other standards the lab were using, or at least work with the labs to make sure that they are standardizing if they are going to be contributing to the database. But if that's not possible in some cases, they'll at least get some sort of alert that there's a discrepancy, and then we can work with those labs to make sure it's curated. Thanks.
During your presentation, you mentioned that data will be made available to researchers. Like, what kind of model you want to provide the data for research purposes? Is it completely de-identified so people have access and can use it in their own environment, or you provide the research environment and they have to use the prescribed tools to do this? And whether they will be able to publish their discoveries and findings.
Well, I can speak maybe to how this is going to be used for Coger. So on one hand, Coger will be used for quality assurance for some of the laboratories. So we won't have de-identified, so we'll have de-identified information into the database. And some of that will be pushed publicly using like the ClinVar submission template. So just, the variant, info about the variant, its coordinates, the submitter, what classification it was given, what standards were used to come to that interpretation. But there may be additional like info on the back end that we're collecting as part of the database, such as like case level data. And in that case, there would be controlled access, and maybe only like lab directors would have access to that level of information. So we are performing needs assessments with all of these different diagnostic labs that I discussed. And we're talking about things just like that, like what would be most helpful for you in terms of using this database, also long-term, what information would be helpful for your own quality assurance and standardization. So all of that will be considered once we're ready to build it out. So, yeah.
Okay, thank you.
Welcome.